related to patients. So preclinical means something which we have to do before experimenting it on patients. So these are the studies which gives the safety and efficacy of a particular drug or formulation. It needn't be drug or formulation alone. It can be a device or it can be a protocol. So anything can be adopted for research studies and anything can undergo preclinical trials also. So we'll see what preclinical trials are. So preclinical trials are also known as non-clinical trials. They're also known as non-clinical trials. That is, they are not oriented to patients. They are not dealing with patients directly. So something which is not dealing with clinics or not, they are known as preclinical trials. And usually we do it either in laboratory setting or we'll do it in animal subjects. Okay, so there are two subdivisions for preclinical trials. It can be either oriented to or stuck on to laboratory setup, or it can be done in animal subjects. And as I have already mentioned, the prime aim of preclinical trials is to establish safety and efficacy of the thing which we are using. It can be a device, it can be a drug, it can be a combination of drugs, it can be a formulation, or it can even be a protocol. So in order to establish the safety and efficacy, we are doing preclinical trials. And this is exactly what I have already told. Products may include new medical devices, drugs, gene therapy solutions. Anything can be included in, or anything can be taken as a subject for research. Generally, they are performed on animals like mouse, rat, guinea pig, hamster, rabbit, cat, dog, monkey, etc. So there are various animals that can be used based on which organ we are going to study, or based on what effect we are going to get. Based on that, we will be selecting the corresponding animals. We'll deal more detail in the coming slides, and also it can also be done on laboratories, as we have already mentioned. It can be an analytical study. Okay, just to know what is the basic active ingredient which is going to give a specific, if we are going to separate an active ingredient, or we are going to detect a specific ingredient, it can be a lead to many other properties of that particular drug. For example, we are going to in research in a particular drug, we found out a particular flavonoid, or we found out a particular active ingredient which is good for antioxidant activity. So we can use that particular drug for studies of any antioxidant activity, that is hepatic antioxidant activity, heart related antioxidant studies, kidney related antioxidant studies, the nerve cells related antioxidants. So in wide areas in that purview, we can use that particular drug. So that is exactly what happens in analytical studies. When coming to animal studies, we usually perform it on these animals based on the effect, or based on the research that we are taking. And with this preclinical trials, what is the ultimate aim? Of course, we understand the safety and efficacy, and also unfavorable compounds, we can delete them from our study, or we can reject them from our study. So these are the main aims or portfolio of doing a preclinical trial. See, this is the drug development process. Okay, this includes the whole of the research. Okay, all the steps involved in the research. So what does we do? We do a preclinical trial. We make a drug discovery, or we take a random drug. The scientists happen to come across a new species, or a new thing, or a new the part of a plant. So what will they want to know the effect of it, or they want to, or just see that if you want to develop a drug which is hypoglycemic, or you want it against hyperglycemia. So we will be randomly choosing some of the a lot of drugs which have hypoglycemic activities, which can be given for a diabetic patient. So we'll be taking around 10,000 compounds, or maybe more than that of compounds we will be segregating and taking. Okay, and then what will we do? We will let it happen. Preclinical studies would be done. So based on this preclinical studies, based on analytical studies, and we'll be seeing the active ingredients, and we will go give it to the animals, and we see the hypoglycemic effect of these compounds in the animal studies. So first, in the analytical studies itself, we will see that if there is no anti-hypoglycemic or anti-hyperglycemic, the ingredients are not present, such drugs will be eliminated there itself. Then we move on to the animal studies. So in the animal studies, again, we will be inducing the hyperglycemia in the animals, and then we'll give this drug, and then we will check on it and see how much of them have safety, how much of them have the efficacy. Okay, so when and we get a few amount of, then again we will reject a few compounds, so that which do not come under these portfolios. So what will we do? It comes under, we will be again doing the preclinical things. Then what will we do? Then we understood around 250 compounds have passed the preclinical trials also. We will then go on to the clinical trials. In the clinical trials, of course, it has got three phases: phase 1, phase 2, and phase 3. Phase 1, we will be doing it in volunteers, healthy volunteers will be doing it. Okay. Next, for those that has passed this phase 1 level, that is no problems has been detected in the volunteers, no side effects seen. Then what will we do? We move on to the phase 2, where we take in volunteers with diabetes, and then we give it. Then what will we do? Then again, and that will be around 100 to 500 only we will check, because again we'll have to know whether there will be any side effects, and we'll be continuously monitoring them on a specific area. Okay. Then we move on to the phase 3 trials, where we'll be giving it widely to the diabetic patients, and they will be let free. Okay, we want to constrain them to hospitals and all. They will be asked to live a normal life taking these drugs, so that we can ascertain that this is safe in a normal living life. This drug is safe for diabetes. So when these three phases have been passed, then this will be sent for FDA review. So such compounds, maybe like we will get only 4 or 5, or maybe one compound only, sometimes we'll get. It will be sent for FDA review, and the FDA will be Food Development Association, that will be assessing the trials, assessing the evaluate the results, and what will they will give consent for making it into a drug, and they will allow them to reach the markets. So again, there is a phase where the phase 4 trials or post-marketing phase, where once it is marketed into the place, or into the public, they would assess it. Okay, they would assess on this the portfolios, conditions, etc., and they would assess whether this is safe for around 5 years, 10 years. They will be continuously assessing whether this is safe for the community, and when it is found safe, then the drug would surpass the phase 4 trials also. It is when in phase 4 trials, when we report complications, ADRs, or other problems, it is then that certain drugs get banned, or the government, the drug controller will ask the specific company to withdraw that specific drug, so that it is causing complications to the public. So these are the different phases of different processes involved in drug development. So know that it is not a simple thing. We know that vaccines are being developed for COVID problems and all. Just see, usually it is this. See, for just drug discovery itself, you will eventually take around 5 years. Preclinical studies, it will take around 1.5 to 2 years. Clinical trials, if you are doing it properly, it would take around 6 years or more. And for FDA review and for post-marketing analysis, it would take another 5 years. So this is not a small process. So even it is just after COVID has come, it's just 2 years it has gone. So it is not very easy for the various scientists to develop a medicine or a drug to surpass this, because it has to go through strenuous trials to make it safe for the public. So this is the procedure for drug development. Next, This is the steps that have been enumerated. You have to identify the molecule or formula, establish the target. That is, we are going to identify a hypoglycemic molecule. You have to establish the target as pancreas, pancreatic beta cells, or whatever thing is. Or if you are going to target to hypothalamus, you have to establish a target and develop the bioassay, corresponding bioassay for doing the preclinical studies. Then you have to screen the drug, establish the safety by animal studies, establish preclinical efficacy also via both these assays as well as animal studies, and then proceed with clinical trials. So these are the steps of drug development. Next, we'll see what screening is. Okay, so screening is a thorough investigation to get pharmacological activity of a new or a chemically undefined substance, and to investigate the functions of the endogenous mediators, measure or define the toxicity of the unwanted actions. So by screening, what all are the points that we would be covering up? We have to know the pharmacological activity of it can be a new drug or a chemically undefined substance. Okay, either we would be like, this is exactly what happens in Ayurveda. So we have certain things written. This is good for Prameha. This is good for Yagrat Shobha. This is good for the Shukrakshaya. So many drugs are being mentioned in our classics. Okay, and we are using it clinically also, without knowing the pharmacological activity of such drugs. So this is one area where we have to investigate on various formulations as well as single drugs. So we know that a particular drug is good for Shobha, or we know that a particular drug is good for Prameha. So what will we do? We have to take that particular drug and understand the pharmacological activity of this particular drug. That is exactly what is mentioned as you have to either get the pharmacological activity of a new compound or a chemically undefined substance. Okay, we know that the specific, amlaki is good for this thing, good for prameha, but we don't know what pharmacological activity of amlaki is giving that hypoglycemic effect. So there, chemically it is undefined substance. And if a modern pharmacy is going to develop a new drug for hyperglycemia, then that is a new substance. So it can be by screening, we are going to get the pharmacological activity of either a new substance or a chemically undefined substance. And then what will we do? We have to investigate the functions of the endogenous mediator. So what happens when you're going to give this particular drug or particular formulation? What is going to happen internally? Inside our body, what physiological variations are going to happen, that we have to analyze. Then we have to measure or define the toxicity or the unwanted action, if there is any side effect, if there is any toxicity on long-term usage, on short-term usage, any problems histopathologically, all these things you have to know. So this is done by screening. And there are three types of screening: sample screening, blind screening, and program screening. Coming to simple screening. So this is done to find the substances having a particular property, to find the substances are active in a single way by simple tests. So this is done. Simple screening is that we use very simple tests to analyze whether a particular drug, we know that amlaki have a hypoglycemic activity. So we'll be using small test. Just we know the property of amlaki, and we have to just test that particular property only. But we are given an unknown drug or a drug with unknown activity. Then we have to we have to screen whether it has hypoglycemic activity or hyperglycemic activity. Or we have to see whether it is having the hypertensive activity or hypotensive activity, hyperlipidemic activity or hypolipidemic activity, whether it is going to give a tachycardia or a bradycardia, or it is going to be neutral to our body. So we don't know what the effect of a particular substance is. Such things cannot be done by simple screening. But simple screening can be done only in drugs with which we know a relative activity. We know that this can be used for this, but we have to confirm it whether this is actually hypoglycemic, and we have to confirm its pharmacological activity as well as its active ingredients. So in such cases, we will do the simple screening because we already know the action of that particular drug. So this is exactly what we do by simple screening. Okay, example, hypoglycemic testing, where ability of a compound to diminish the blood glucose level will be tested. So this is simple screening. Now, next one is blind screening. Blind screening is what I have, as I have already told you, we do not know for what we are going to use a particular drug, for what we are going to use a particular substance. So that is blind screening. And new chemical entity or isolated naturals will be given to us, and then we have to test on, we have to make many blind tests, and then we have to come across the, or we have to conclude on the property of that particular drug. Okay, then only we have to think whether it is worthy for further attention. It very much requires planning and skillful execution. Nature, economical, time, money, etc. Everything has to be looked into. Toxicological screening is also necessary. Next, it is programmed screening. So programmed screening provide detailed information on activity, addresses how, why, and what on activities. Okay, so here we are giving, by programmed screening, we are going to give a detailed information about that particular drug. It will investigate the primary action, secondary action, subsidiary activity. Everything will be given in more precision. So that is what is meant by programmed screening. That is investigated for some particular pharmacological activity which is system oriented or organ specific. Okay, so that even the clear pathway of action would be given by programmed screening. So this is a detail. So after simple and blind screening, when we evaluate that, yes, this is having this particular property, it is having a hyperglycemic or hyperlipidemic activity. Okay, then we will do, how is this causing this hyperlipidemic activity in the body? That pathway will be cleared by the programmed screening. This is exactly what is meant by pro. So we have three types of screening: simple screening, blind screening, and programmed screening. In simple screening, what happens? In simple screening, we know the relative action of the drug, so we will be just confirming whether this is having this particular activity or not. In blind screening, we do not know the activity of that particular drug. It is a new drug, so we will be randomly checking the particular activities, what all activities that particular drug have. And when we analyze and when we conclude on this activity, whether it is primary, whether it is simple or blind screening, the drug will be subjected to a program screening where we will be understanding the specific pathway of that particular drug. So this is by program screening. These are the three types of screening. Now, regarding the types of studies. Okay, so what are the types of preclinical studies? So we have already seen that preclinical studies have such and such objectives, isn't it? It is used for safety and efficacy. It is used for knowing the pharmacological activity, whether it has to be considered. For all these things we are using, and it is also how we do the basic the theory of doing preclinical studies is to do screening, isn't it? So we saw what screening is also. Now, what all studies are used for this screening? This is in vitro study is there, in vivo study is there, and in silico studies. There are three types of studies come under the preclinical studies by which we do screening. Okay, so what are the three types of studies? It is in vitro study, in vivo study, and in silico studies. Coming to the in vitro studies. So what is in vitro studies? These are relating to cells. In vitro, vitro means inside a cell, so inside a closed environment. So these are usually cell-based studies. It can be antibacterial, antimicrobial, antifungal assays. We are using, with the aid of assays, we will be understanding the particular action of a drug, whether it is antibacterial, microbial, antifungal, cytotoxic, receptor, is it doing a receptor binding, is it causing some enzyme inhibition, DNA, RNA, PCR test, antioxidants. So by using various assays, okay, microbiological and biotechnological assays, we will be ascertaining the pharmacological activity of a particular drug refined to or confined to a particular cell. Okay, so this is the basis of in vitro studies. If we want to understand the action of a particular drug in the heart, we will take the heart cell lines. If we want to understand the effect in the pancreas, we'll take the pancreatic cell lines. So the corresponding cell lines would be taken, and this primary study would be done in order to establish the efficacy or establish the activity of a particular drug. So this is a primary study involved. Next is in vivo studies. So what is in vivo studies? They are pharmacological studies to establish the efficacy. Okay, so what is meant by in vivo? That is in relation to the animals. In vivo studies are in relation to the animals. So pharmacological studies to establish efficacy, analgesic, antipyretic to understand analgesic activity, antipyretic activity, etc. We use in vivo studies, and then will be used for toxicological studies to establish the safety, for understanding the dose conversion and dose determination, for cumulative effects. So four properties or four effects can be analyzed by animal studies or in vivo studies. So what are they? They're pharmacological studies to establish the efficacy. We can understand the safety by undergoing toxicological studies in animals. We can know the dose, corresponding dose that has to be given to the humans by dose conversion, by giving it to by giving and understanding the efficacy and safety. We can convert it into human dose. So for dose determination and dose conversion, we will do in vivo studies. And to understand the cumulative effects, that is, if you are using this particular drug for 6 months or 1 year, will it cause any cumulative effects in the body, whether the excretion, metabolism, digestion, ADME of the drug is normal or not. So to understand these properties or understand these points, we will be doing animal studies. Okay, pharmacological studies is example is analgesic, antipyretic. Toxicological studies, you have to know the LD50, acute toxicity, subacute toxicity, bioavailability of the drug, plasma concentration, histopathology. For all these things, we will do the toxicological studies. Then it is dose conversion and cumulative effects. So these are the aims of doing an in vivo study. Next, in silico studies. So what is in silico studies? In silico is an expression performed on computer or via computer simulation. That is, it is an alternative to animal experimentation. Okay, that is the expression in computers. We'll be feeding it an expression. Not much researchers have gone in silico studies, so just very primitive knowledge only we get with in silico studies, because we all know that programmed and computerizations, we are all biological living things. So only when we actually give and see it in animals or see it in living organisms, we can actually understand the safety or efficacy of a particular drug. But programming it and doing it in computer-based things and all can always limit the knowledge given. But still, we can actually use this types of studies to screen, actually, in order to screen whether we have got some 5,000 compounds or 5,000 drugs are given, and we have to screen it. We have to take only the best 100 drugs. In such cases, we can rely on in silico studies rather than using these 1,000 drugs in animals in cell lines. And rather than that, in such cases, we can use in silico studies where screening is relatively easier. So this expression in silico was first used in public in 1989. So this is about in silico studies. So these are the three types of preclinical trials or preclinical studies. Now we'll move on to experimental pharmacology. So actually we understood that by preclinical trials, we have seen that around 20 or 50 drugs are good for the particular activity that we are going to do a research. So the next phase is that we have to go on for an experimental pharmacology. So how do you do experimental pharmacology? That is, analytical studies will be there, and also clinical, experimental, and pharmacotherapeutic pharmacology will be there. These are the subdivisions of pharmacology. Pharmacology is study of drugs. The definition of pharmacology is study of drugs. So basically, the study of drugs can be divided on the basis of analytical studies, as well as the next session is clinical, experimental, clinical, and pharmacotherapeutics. Analytical studies, actually the QC parameters of a particular drug will be analyzed. Okay. And in experimental pharmacology, the effect of various pharmacological agents would be studied on different animal species. Again comes the animals here, experimental pharmacology. There we are like just using it for understanding a base idea. Okay, basic idea we use in vivo studies. Here in experimental pharmacology, we'll be like using it much more deeper, like we will be using it for some 6 months or maybe more than that in order to evaluate the correct action in a particular animal. So these are the types of pharmacological studies, and aims of experimental pharmacology is find out the therapeutic agents suitable for human use, study the toxicity of the drugs, study the mechanism and site of action of the drugs. So analytical studies. First, we'll see about what is analytical studies and what are the parameters that we have to check in analytical studies. It was actually in 1998, WHO has published quality control methods for medicinal plant materials. And at the end of 2007, WHO guidelines were documented for assessing quality of herbal medicines with reference to contaminants and residues. And it was also published in 2007. And it was in 2010, as an annex one of the 44th report of committee, actually the WHO expert committee made specifications for pharmaceutical preparations and adopted it in WHO good practices for pharmaceutical quality control laboratories. So it was in 2010, actually it was well properly documented for herbal drugs to follow QC parameters while production. And the basic criteria is identity, purity, and assay. You have to check the identity, you have to check the purity and the assay. Okay. Now coming to the different kinds of analysis of QC parameters. What we have to do is that when we get a drug or a formulation, you have to make the standardization. You have to make the quality evaluation of that particular drug or formulation. So what are the parameters that we have to check? We have to check the physical parameter, botanical parameter, biological parameter, and chemical parameters. In physical parameters, moisture content, ash value, extractive value, viscosity, density, bitterness value, solubility, swelling index, forming index, specific gravity. These are the points that we have to check in physical parameters. In botanical parameters, macroscopic and microscopic parameter. Macroscopic parameter, color, order, taste, texture, and fracture. Microscopic, qualitative, quantitative, SEM studies, powder studies. These are the parameters that we check. In biological parameters, microbial contamination, pharmacological evaluation, toxicological studies have to be done. In chemical parameters, chromatographic, heavy metal, pesticide residues, and mycotoxins have to be checked. So why are we doing all these parameters? Why these parameters have to be checked? This is that first point is that we have to understand whether if it is an unknown compound, we have to know whether the particular drug we are going to take is of value, is going to make any valuable contribution to our study. Is it going to give us any effect? So it is firstly for screening. Secondly, when you know that a particular drug is having a particular quality, okay, when you know that Amlaki is having a hypoglycemic activity. So then why are we doing the analytical studies? In that case, we have to do these analytical parameters in order to confirm its purity. Okay, in order to confirm its purity, as we have already mentioned, identity, purity, and strength. These are the three parameters that we have to always look in drug development. First of all, you do the analytic parameters to identify a particular pharmacological activity. If a particular activity is identified in a compound, then we will be doing it again and again. In each time when you are taking amlaki, we will be doing this QC in order to confirm its purity. Okay, so that is and purity and strength also, how good it is. To confirm it, we have to do these analytical studies. So these are the parameters that we have to check for identity, purity, and strength. Next, detailing going to the experimental pharmacology. You know that experimental pharmacology deals with animal studies. Okay, why animals? Why we are choosing animals for such studies? Why not other organisms, not reptiles? Not why are we taking animals? Why don't we do it in humans itself? Because it is having a similarity. For basic cell structure, organ system will be similar. When you take the heart of the frog and heart of the for humans, it will be like its contractions and all are similar. Then we are going to check about the various like hormonal variations in a particular when you take a particular drug in a mice. Then that is because that hormonal action is similar. It is more or less comparably similar to the humans. That is why it is very important for us to choose the correct animal for what study you have to take, for what action you want. Correspondingly, you have to take the animals. If you want to prove, or if you want your study is in relation to something related to hormonal involvement, then you have to take an animal model which have more or less similar action to the human physiology in relation to that particular hormone. So that is very important when you are going to take an animal model. So the basic cell structure or organ system or anatomy should be comparatively similar to the animal that you are going to take, which is comparable to the humans. And also the physiology, the functions of the that particular animal should be comparable to the functions of the human also in regard to the study that you are going to do. Okay. And also to advance scientific understanding, so that you can, like, after the study you can do dissection, you can know the histophysiology, and also you can understand what all has happened in the body after you have given a particular drug. So for all these things, you choose animals for experimental pharmacology. Next, experimental animals. So what are the experimental animals? Rodent mammals. That is rat, Wistar rats or Sprague-Galway rats, long events rats, sucker rats, biobreeding rats, hairless rats, mice. All these are experimental animals that comes under rodents. Non-rodent mammals are rabbits, monkeys, dog, cat. Other animals include zebrafish, frog, and all. Okay, so these are the usual experimental animals that we take for experimental pharmacology. And division of models based on how diseases occurring. Based on that, we can divide them into natural models and induced models. In natural models, the experimenting animal will naturally have a disease which is similar to what we are experimenting. Induced models is that we are taking the animal and we are inducing a particular disease into the model, and then we are doing the study. So these are the difference between natural models and induced models. Now coming to rats. So rats is study why, in which all conditions do we use rats? That is what we are going to see. Rats is study of analgesics and anticonvulsants. Then you want to do a study on analgesics, that you want to know how good it is going to reduce the pain. Okay, you have a pain in the paw, or you have a pain in some organs, and you want to know the analgesic effect, or you want to test the analgesic effect uh, of a particular drug that you are going to give in comparison to the diclofenac. Diclofenac is a standard pain reliever in modern medicine. So you want to compare the effect of the Shallaki or Gugulu in comparison to diclofenac. So then such cases, it is a study of analgesics. In such cases, you can choose the model as a rat. Okay. Or if you want to study the action of in Kupilu, you want to study the anticonvulsant action of Kupilu to the anticonvulsive drug which is compared in modern medicine. Then you can take rat as the model, because your neuroendocrinology is more or less comparable of rat and humans, and neuroendocrinology is comparatively comparable. So we can take rats. Then study of estrous cycle, mating behavior, lactation, gastric acid secretion, hepatotoxicity studies, study on mast cells, bioassay of various hormones such as insulin, oxytocin, vasopressin are all, in all these cases we can take rats as the model. But one thing which we have to be careful and we have to consider while taking rats is that rats are better at removing toxins from their bodies than humans. So it may be possible to refine the use of rats in tox. So if you really want a toxicological study, then it is not recommended for you to take a rats as your study model. Now it is different rats. Different rats is Wistar rats. Wistar, these are developed at Wistar Institute in 1906 and is notably the first rat developed to serve as a model. This was artificially developed for study purpose. And it is currently one of the most popular rats used for laboratory research. It is characterized by its wide head, long ears, having a tail length that is always less than the body length. So this is about Wistar rats. Next, Sprague-Dawley rats. These rats are typically have increased tail to body length ratio as compared to visitor rats. Long Evans rats utilized as a multi-purpose model organism frequently used in behavioral and obesity research. So it's very important for behavioral and obesity research. The rat which is used is Long Evans rat. You also have sucker rats, which is genetically modified rats used to study obesity and hypertension. So for that is for lifestyle modified diseases, you use sucker rats. Well, for behavioral and obesity diseases, you use long Evan rats. Biobreeding rats. Models to study autoimmune type 1 diabetes, you use biobreeding rats. Hairless rats to study compromised immune systems and genetic kidney diseases. Okay, so for autoimmune type 1 diabetes, you use biobreeding, while for compromised immune system and genetic kidney diseases, you use hairless rats. Now, what are the advantages of using these rats? It is small in size, and the drug required for the study is also relatively less. Vomiting center is actually absent, so oral administration is easy for rats. Gallbladders, tonsils are absent for it. Continuous flow of gastric and bile. This facilitates the study of drugs acting on bile, cholesterol, reabsorbing, etc. So, if you really want to understand the pharmacological activity based on gastric mucosa, intestine, anti-allergic, bile related, in such cases is the best model that you can rely on is rats. Next, it is mice. Okay, so mice are actually used for toxicological study because we have already seen rats have relatively more ability to eliminate the things from toxicity things from the body. So toxicological studies, rats are not that good, but compared to rats, mice are better for toxicological studies. Teratogenicity studies, bioassay of insulin, screening of analgesic, anticonvulsant, screening of chemotherapeutic agents, that is very important, screening of chemotherapeutic agents, studies related to genetic and cancer research, drugs acting on CNS. In such cases, mice are better. Advantages of mice is that it is the smallest of all the models, easy to handle, cheap, and less prep is required. Swiss albino mice are the commonly used species, and other strains are Balbarsi and C57 strains. Now, coming to the basic difference between rats and mouse or rats and mice is that size and weight of the rats are more than that of mouse. Mouse has a pointed face compared to the rat. Rat has a thick and heavy tail while mouse has a thin tail. Rat has 22 pairs of chromosomes while mouse has only 20 pairs of chromosomes. Rats have longer gestation period compared with the mice. Okay. So if you want to understand the teratogenicity, that is exactly why you use mice for understanding teratogenicity studies. You want to know the teratogenicity activity of a particular drug that you wish to give for a pregnant lady. Then it is better to use a mice because it has a shorter gestation period than the rats. So you can easily get the result. Okay. So for teratogenicity studies, we rightly use mice rather than rats. These are the differences of rats and mice. Now coming to the next set, that is guinea pigs. We have seen rats, mice of the rodent one. Now coming to the next set of the rodents, guinea pigs. They are evaluation of best evaluators for bronchodilators, anaphylactic immunological studies, histamine, antihistamines, bioassay of digitalis, evaluation of local anesthetics, hearing experiments because of its sensitive cochlea, isolated tissues, especially ileum, heart, etc. Study on TB, ascorbic acid metabolism. So for all these studies, the best model that you could rely on is guinea pig, especially for antihistaminic activities like allergy-related activities, immune system-related activities. Such studies, the best option is guinea pigs. Next, rabbits. Rabbits are best for bioassay of anti-diabetic sex hormones, screening of agents affecting capillary permeability, pyrogen testing, drugs used in glaucoma, pharmacokinetic studies, studies on local anesthetics, studies related to anti-fertility agents, isolated preparations like heart, duodenum, ileum. So in such cases, so something related to the genito-urinary system. In such cases, rabbits are the best option. Next, frogs. Now usage of frogs is actually banned now. So Just understand. Usually we don't use frogs for any studies now, but understand its utility. Commonly used in physiology, pharmacology, and toxicology. Isolated preparations, heart, rectus, abdominis muscles, such studies when it comes with frogs are a better option. Drugs acting on CNS, NMJ, so neuromuscular junction, that is NMJ. So such studies we prefer frogs, but now it is not, it is ethically not allowed. Next, monkeys. Monkeys are more or less similar to humans in emotional behavior, menstrual cycles. So they are usually used as models for virology, parasitology, immunology, fertility, nutrition-based studies, etc. Especially for COVID vaccines, the model was monkeys only. Dogs. So the next is dogs, and they can be easily trained for behavioral studies. So always remember, for behavioral studies, the actually the model is dogs, and they usually follow oral command also, and suitable for antihypertensive studies, vaccination studies, diabetes, ulcer experiments, etc. So these are the concerns, or these are the informations regarding the experimental pharmacology, and they revolve around animals. Okay, we are doing experiments on animals, and we are getting maximum knowledge out of animals regarding the safety as well as efficacy of a particular drug. So that is experimental pharmacology. Next, it is clinical pharmacology. Clinical pharmacology is trials on humans. It is actually, we always do it on healthy volunteers first. It is also used to assess the safety and efficacy, comparable with phase 1 of clinical trial. Comparative trials with known drugs, dose determination is made by clinical pharmacology. ADR and ADE can be elicited by clinical pharmacology, and overall it aims to generate data for optimum use of drugs and to create evidence-based medicines. Next, regarding the pharmacodynamics. Okay, so regarding the pharmacodynamics of a particular drug, it is uh, what we are going to do is drug action, drug effect. That is what we are going to analyze. Okay, and the principles of drug action, mechanism of drug action, and combined effect of drugs. So these are going to be evaluated in pharmacodynamics. First, we will see about the definition of pharmacodynamics. Okay, so pharmacodynamics is a study of the drug effect, what a particular drug is going to do on our body. It is going to, it is the study of drug effects. It explains the complete action effect sequence and the dose effect relationship added with modification of action of one drug with another. Okay, so what it explains is completely, it is going to give us an idea about the action of a particular drug on our body and effect that is going to give us. And it will also give us the modification, and with it modification of action of one drug. But if you're going to give two drugs together, then how one drug is going to modify the effect of the other. Whether it is going to have a synergistic effect, whether it is going to have an antagonistic effect, or whether it is going to have a neutral effect, that is going to be evaluated by the this thing by the pharmacodynamics. Okay, so synergistic is that add-on effects, and antagonistic is that lessening effect, and neutral effect is neutral effect. So these are being understood by pharmacodynamics. Now we will see what drug action, drug effect is. Drug action is the initial combination of the drug with its receptor in a conformational change in the latter. Okay, in case of agonist, or prevention of conformational change through exclusion of the agonist in case of antagonist. So for this, you have to understand the actually how a particular drug is going to act. Okay, always there will be a receptor and there will also be the drug. Okay, so this drug is going to come and it is going to actually it will go to many sites only when its head is going to lock. It is a lock and key procedure. A particular receptor will have a area where the this particular thing, the drug compound or drug active principle will have a head also. So this head should get locked inside the hole or the key of the particular receptor. So it is rightly when it comes and gets locked, actually a particular drug action gets elicited. And the drug action can be either, it can be again an agonist or it can be an antagonist. That is, if a particular drug is going to come log and is going to initiate an action, then it is going to be an agonist. If it is going to like change it, that is, you have a fever and this is going to come lock it, and this is going to give a antipyretic effect, then it is an agonist. Antagonistic effect is that you have a, like you have an increasing production of of thyroid hormones. T3 production is very high. Okay, and when this is going to come, when a particular drug is going to come and is going to bind to a particular receptor site, and this is going to decrease the type T3 hormone in your body, then it is having an antagonistic effect. So this is the drug action drug effect. So a receptor, a drug and the receptor combination can be either agonist or can be antagonist. That is, either can be promoting or it can be diminishing. So based on the action of that combination, we can divide it into agonistic activity and antagonistic activity. Next, drug effect. Drug effect is the ultimate change in biological function brought about by the consequence of the drug receptor combination. So, what the body is going to happen? What is the physiology of that is going to happen in the body based on this this promotion, or based on this the mediation? So that is drug effect. So this is drug action, drug effect. So drug action is that it is the what kind of action the